chapter. So we understood that a lot of innovations and a lot of curiosity led to people's observation with respect to animal cell, plant cells, as well as many microorganisms. They were observing it under the microscope, and all of them were evaluating their own perceptions and their own facts about the cells they observe, and they were coming out of this. Um, they were coming and telling their opinion about, okay, this is what we think that the cell is. But today we have a very strong and very convenient knowledge about what we talk about, what we know about cells. With the development of microscopy, now it gives us a tremendous amount of capacity or technology and ability for us to really go deep into the functions of cells. Okay, so coming to what is a cell can be of any size, or any shape, or it can be present in any form. So coming to the size of the cells, so mycobacterium is the smallest cell that is known to mankind, and then your red blood cell is very small cell that is found in your body. Your nerves are the longest cell in your body. Ostrich egg cell is the largest cell that is present on the planet. So now they change in the size. The size varies tremendously. They come in very tremendous amount of sizes. Next one, coming to the shape, the cell cytoplasmic volume, or the amount of cytoplasm it has, the position of the nucleus it has, the number of mitochondria it possesses, or whether it has a flagella or a cilia, whether it is a mobile cell or an immobile cell, whether the cell is participating in an immune function or a non-immune function, or is it a transporter or a non-transporter. You see, cells have so many functions that we can just list down. All the activities that we can think about in the biological system is conducted by the cell and within the cell itself, right? So that is why the cells, or the organelles present in the cell totally depend upon the cell function itself. Okay. Now coming to a general view of a cell. A generic view of a cell would include a cell wall, and then a plasma membrane and a cytoplasm. Correct, right? So now, as we studied about the contributions of Sheldon and Schwann, we also understood that the differences between the Sheldon's observations and the Schwann observations, as the proposed cell theory. Now, Sheldon was the one that observed all the plant cells, and Schwann was the one that observed all the animal tissues. So he came to a conclusion that the presence and absence of the cell wall is clearly identified in a plant cell and animal cell. Similarly, as the observations with the cells progressed, people did understand that there are two levels of organization with respect to cells. One, which is a very primitive model of cell, and other one is a very advanced model of the cell. So, based on the cellular complexities, in simple terms, the presence of membrane or non-membrane bond organelles, we can classify cells as two types of cells. One is a prokaryotic cell, other one is a eukaryotic cell. Now, a typical prokaryotic cell. Usually, we have understood that in biological classification, that we have divided the entire living organisms into five convenient kingdoms: kingdom Monera, Protista, Fungi, Plantae, and Animalia. Now, when we talked about Monera, we talked about prokaryotes. Okay, so Monera includes bacteria, and the bacteria, what we call, right? So the bacteria basically is classified based on the shape it takes. So a prokaryotic cell comes in these many types of shapes. One is round, other one is rod shaped, next one is spiral shaped, and then we have vibrio. Okay. So based on the shape of the cell, we have classified the bacterial cell into a round shaped cocci, then sorry, and then rod shaped bacilli, right? And then spiral shaped spirillum, and then we have Vibrio. Okay, Vibrio shape, Vibrio. See, now the thing is, based on the shape, we are able to classify that these are the different types of bacteria. Okay, so apart from the shape of the bacteria, all these bacteria, they have generally a thick layer that is protecting them. Okay, this we call it as bacterial, um, the protective layer of the bacteria, or the bacterial capsule. See, the protective layer of the bacteria, that means the layers that are within the, see, for example, if you have the plasma membrane, and the layers that are above the plasma membrane act as a protective layer in bacteria. So now we're talking about the protective layer in bacteria. It is usually made up of three layers. What it includes: capsule, a cell wall, and a plasma membrane. So they envelop the entire cell. Now within these layers, what sits is a cytoplasm, which is an uniform matrix where all the cellular activities take place, such as transporting substances, biochemical reactions, okay, and then giving space for the organelles to stay, okay, and then establishing the communicating links between the organelles. So most of the thing, uh, the most important point about the cytoplasm is this homogeneous semi-solid structure is very, very important for giving cell the shape, cell the structure, as well as the medium for undertaking all the cellular activities. So that is about cytoplasm. Within the cytoplasm of the prokaryotic cell, what we can find is very peculiarly, okay, one of the unique characteristics of prokaryotes is, see, we can find that they do not have any organ membrane bond organelles. So there are no membrane bond organelles. So when I say that there are no membrane bond organelles, that includes your plastids such as chloroplast, chromoplast, leucoplast, they are not present. Mitochondria, they are not present. Okay, or any of the membrane bond, like for example plasma membrane, it's not present. So no membrane bond organelles are present in a prokaryotic cell. Apart from the membrane bond organelles, what else is present? Within the center of the cytoplasm there is a dense nucleus, and this nucleus in a prokaryotic cell, especially if you look at a bacterial cell, what you can see is the nucleus is very naked. That means it is very exposed. So what do you mean by naked nucleus? If I take an eukaryotic cell or a human cell, let us say, if I take a human cell, what I can find is my nuclear material is very well protected, right? So this is my cell. So here I have a nuclear membrane, which is a double membrane bonded organelle, where all my genetic material is enclosed within this. So what is that mean? We know that the nucleus is a very important part of the cell, and it controls majority of the cellular activities, all the activities, by telling or giving the information for the cell organelles or the neighboring cells what to do. Now, this information that is present in the nucleus is is in the form of genetic information, which is popularly known as DNA. You might have heard about DNA, or you might have heard about this term called DNA. If you don't know about DNA, don't worry, you will learn about DNA very much detail in the coming classes. Okay, so the information which is controlling all the characteristics, for example, what is characteristics? Something you can see and differentiate, or something it gives its uniqueness, a property that is called as a characteristic. Correct, right? So, as a human being, or as any organism that you can see around, we express a lot of characteristics, not only behavioral characteristics, even the way the food we, the way we digest the food, the way we transport oxygen, the way we utilize oxygen. Correct, right? As a living organisms, we have so many, activities to perform, and these activities can be just called as a characteristic of an organism. So these characteristics are very well stored in the form of DNA, which is a genetic information. This DNA is further packed into the within the prokaryotic and eukaryotic cells. So, if you see how the DNA is present within the prokaryotic cell, you can see that there is a single circular DNA which is double stranded. So there is the DNA which is present in the bacteria is circular and it is double stranded. Okay, so they represent chromosomes. Now, if you see the bacterial cell, because they don't have the nuclear membrane, their genetic material, the very important information, is quite naked. It's exposed. It's not well protected like us. So that is what is the major difference when it comes to prokaryotic cell and eukaryotic cell. So now, since the DNA is exposed and it is not within the membrane, so that region where the genetic information is stored is called as nucleoid. So the genetic information of the prokaryotes are not called as chromosomes; they are called as nucleoid. Okay, so that is how they have been organized into. Okay, apart from that, there are certain structures we will discuss about it and how it sets this prokaryotes quite different. Now, let us take one organelle. We just took a very general view of the prokaryotic cell. Now we'll go a little bit detail. Now the prokaryotic cell, like any other plant cell, now what you can see is there is the cytoplasm. The cytoplasm of the bacteria or the prokaryote is very well enveloped by something called as cell envelope. So the cell envelope is basically made up of three tight bound layers. The outermost layer is called as glycocalyx. Next one is a cell wall, and below that is the plasma membrane. Now they have their own chemical composition, and then they have their own role to play. Okay. Now coming to what is this? Okay, sorry. Coming to what is glycocalyx. Glycocalyx layer is what do you tell? It's a very um, mucilaginous sheath that is present outside the cell wall. Based on the type of the glycocalyx that is present, the glycocalyx can either show a very soft and very slippery appearance, or sometimes the same glycocalyx layer, it can look very rough. So that is how we differentiate the bacteria based on the type of glycocalyx that is present, whether it is slimy or whether it is rough. Now, why this is important? Again, we can see that there is little bit of characteristics that is shown in the bacteria. That slimy ones are mostly the disease-causing ones, and the rough ones do not cause disease. Okay, so that is one reason. So this is about glycocalyx. So the another layer is the cell wall. The cell wall of the bacteria is a very important uh, area of interest. Why? Because even though the bacteria's cell wall is made up of murin or peptidoglycan, okay, it is made up of murin or peptidoglycan. So now when I say what is it made up of, for example, if I tell you, if I ask you a question, what is the cell wall of a plant made up of, what you will tell? You will tell it is made up of cellulose. Similarly, if I ask you a question about what is the cell wall composition of a prokaryote or a bacteria, it is mostly made up of murin or peptidoglycan. Okay, so peptidoglycan is made up of protein and sugars. Now what happens is this peptidoglycan, it comes in two ways. One, based on the amount of peptidoglycan present, we can differentiate this prokaryotic cell, into whether it is gram positive or gram negative. Let me not confuse you. What I'm trying to say is, I told you that the capsule of the prokaryotic cell is made up of the outermost glycocalyx. We understood the nature of glycocalyx. And the next layer, sorry, and the next layer we have to study about is the cell wall. When I tell the cell wall, first what you have to tell is the composition of the cell wall. The composition of the cell wall is made up of peptidoglycan or murin. Now, based on the composition of the cell wall, it allows us to divide or to understand bacteria into two important groups. So to understand them into two important groups, we have to perform a particular staining technique. Staining means you just mark it with something. So that stain is called as gram staining. Okay. So what do we do in gram staining? Nothing. Actually, we are doing something, not nothing. So we are taking a bacterial cell and then we are trying to label it with a gram stain where we use crystal violet. iodine, and then we use or sometimes we use you know, methyl orange or something like that. Okay, finally, by the end of the gram staining, what you can see is either you can see blue color or you can see pink color. Okay, because of saffronin, not methyl, but saffronin. See, the reason why two different bacteria appear in two different colors. Why blue? Why pink? The ones that appeared blue is because they had large amount of peptidoglycan and the gram stain was, you know, held or bound very tightly to the cell wall. So if they are bound very tightly to the cell wall, that makes them blue, that makes them gram positive. Okay. Now, the ones which have very less peptidoglycan layer, they cannot hold, uh, crystal violet stain or any gram stain. So now what it happens? It does not hold the stain. Now it starts appearing pink. Now we call them as gram negative. Now, why was this important? This revolutionary staining technique that allowed us to differentiate between gram-positive bacteria and gram-negative bacteria ultimately led for the understanding that gram-negative bacteria are the common culprits for causing disease in many of the organisms, whereas gram-positive bacteria is known to be found very beneficial for human beings. Okay. Now, coming to the plasma membrane of the prokaryote is very similar to that of eukaryote, and we will study the properties of the plasma membrane in the later class today. Now, one more important fact about uh, the membranes or the cell envelope is if you look at the plasma membrane region of the bacterial cell. Okay, are you guys understanding? Are you following? Students, are you able to understand? Okay, just say hello. Are you reacting? Are you there? No. If you understand, say yes. If you don't understand, say no. No reaction. Okay. If you have doubts, let me know in the chat box. Okay, and communicate. It's okay. You have to just let me know what your concerns are. If you're understanding, is my voice audible? Can you see the presentation properly? All that is very must. Okay. So just let me know. Okay. Thank you for the response. So, so now talking about the plasma membrane, there is some unique um, region in the plasma membrane of prokaryotes. What you can see is if you see the plasma membrane of an eukaryote, it's like this. Whereas if you see the plasma membrane of a prokaryote, it's not like this. This double membrane bond is quite folded. Okay, so it's folded into inner folding, and these inner foldings sometimes have vesicles, sometimes have tubules, and sometimes they have lamellae, something like this. Okay, so something like this. So vesicles are bubbles, tubules. See, you can just understand this. These are vesicles, okay, and these are tubules, something like this. Okay, tubes, pipes, okay, and then you have vesicles, tubules, and laminae. Laminae would be, you see, the thicker at the end, thinner at the center, and they're either concave or convex shape. Okay, so that is how they are organized, and they are very very important bodies. So these inner foldings you can see here, this is a plasma membrane, and this inner folding is very very important. Why this inner folding is important? These regions in the inner foldings of the plasma membrane, we call them as mesosomes. Now, what is the purpose of mesosome? Mesosome has several functions. One of the major function of the mesosome, okay, what it does is it helps in, okay, preparation of energy. Okay, so how does it help in manufacture of energy? Mesosomes are actually like mitochondria. Okay, I'll write it this like mitochondria. Okay, their job is to perform like mitochondria and provide energy for the cell. First thing. Second thing, what is the other function of mesosomes? It helps in the manufacture of cell wall. If the cell wall has to be manufactured, they help in that. They help in cell division. Okay, so they help in cell division, they help in manufacture of cell wall, they prepare energy, okay, like that of the mitochondria. Understood? So this is about mesosomes. Now, coming to the vesicles, so basically they also have certain chromatophores, where they have chrome containing or pigment containing membranous inner foldings. So example, cyanobacteria. You see some of the bacteria who are photosynthetically active, they have certain vesicles where they have the pigments which can produce just like which can act like a photosynthetic pigment. Understood? So this is about the chromatophores. Now, we understood that in our prokaryotes, we have a cell wall and we have plasma membrane. And in plasma membrane, we came across a structure which is called as a mesosome, which is very similar to that of our mitochondria. Correct, right? It manufactures energy for the cell. It helps in the manufacture of cell wall. Okay, and it also helps in the cell division. So these are some of the functions we picked up with respect to mesosomes. And also, these vesicles, they can be either reserve food material, or it can be waste, or sometimes it can be pigments in case of some of the photosynthetic bacteria such as cyanobacteria. Now, coming to the other important factor is the cytoplasm. So, as I told you, the cytoplasm of a prokaryote is a very homogenized, semi-solid, a structure. So now, what happens? Imagine this is a room. Imagine this room. In this room, what we can see is there is air, right? So air is a mixture of particles that is helping us get oxygen. It is giving us the temperature. It is giving us environment. Okay, everything depends upon what is present around us. Similarly, the cytoplasm provides such an environment where all these cytological activities can take place without any disturbance and without any damage to the cell. Understood? Within the cytoplasm, as I told you, we have our genetic material which is present at the center, and they do not have any membrane. So it is a non-membrane bond structure. Okay, the nucleus of the prokaryotic cell is non-membrane bond. So this non-membrane bond structure encloses, or within that, what we can find is circular genomic DNA. Now I told you what is DNA. DNA is a genetic material. Even we have genetic material. All our cells have genetic material. Okay, we'll talk about it. So this genetic material is have to be arranged. So the arrangement is in the form of circular fashion, like a bangle. So DNA actually is like a chain. Okay. So what happens if you just meet two ends, it becomes a circle. So that is how the DNA of a prokaryote is present within the cell. Understood? And they also have several proteins which is helping this DNA to pack. Now, there is some concept, and this concept is called as extra chromosomal DNA. What is extra chromosomal DNA? Usually, what happens is when you talk about a cell, the cell's genetic material usually it lies within the nuclear region. Within the nuclear region, we should have all the genetic information that is stored. But sometimes what happens out of the cell also, you can find some of the genetic information. Now let's say that apart from the nucleus, away from the nucleus, if we have genetic material, can we consider it as a genetic material of significance? We can only give importance to the genetic material which is outside the nucleus only and only if it participates or it gives a specific character to that particular uh, organism. Now, for example, we call one of the genetic material which is outside the nuclear region. So this is called as a plasmid DNA. Okay. So a plasmid DNA is an extra chromosomal DNA. So what do we call it as? Extra 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 chromosomal DNA. Now, why do we call it as extra chromosomal DNA? Because it is not within the it is not within the nucleus, right? It is outside the nucleus. That is why we call it as extra chromosomal DNA. Now, why do we have to study about plasmid? Before we understand that, how does plasmid look like? Plasmid also looks circular, but it is double stranded. Double stranded means what? Two chains are present. One chain and two chain. In biomolecules, we understood, right? What is double stranded? So this is double stranded. So there is double stranded circular DNA outside the nucleus is called as a plasmid DNA. Now, plasmid DNA is a very significant DNA for us. Why it is highly significant for us? For several reasons. One of the major reasons is we use this plasmid DNA for many of our biotechnological studies and genetic engineering. Anyways, now coming to the cell and its functions, this plasmid DNA, why we study is when you look at bacteria, bacteria, unlike any other organism, it lives in an environment. And we know that bacteria has a very bad habit. What is a bacteria's bad habit? It goes and infects organisms and gives it disease. It does the same thing not only to us but any other organism as well. So, like us, other organisms don't keep quiet, they fight back. Now, let us take an example. We have a fungus. Okay, we have a fungus. Now, this fungus is living among bacteria. So now what happens is this bacteria is trying to infect the fungus. When it is trying to infect the fungus got infected and it unfortunately died. Now the next generation of the fungus that is coming is very aware that there is this specific bacteria and if it attacks us, we will definitely die. Like how we understand disease, same way other organisms also understand. Now this younger generation is very smart. What they do is we are not going to surrender and die. Instead, we are going to create some poison that can kill bacteria. So what they do is they prepare some poison which can kill bacteria. This we call it as antibiotics. Okay. So can you tell me who isolated the first antibiotic or who discovered the antibiotic? Let me see your answers. Who discovered the first antibiotic? It's a easy question, students. I know that. Come on. You have to give me an answer. Who discovered the first antibiotic? Don't know. Or you know. Okay, I'll just wait. Okay. Just type your answer. Who discovered the first antibiotic? It's very easy. You might have heard about it a very lot of times. And I know that you know the answer. Just type it. It's fine. Okay, if it's wrong, don't worry. All right. So yeah, I was talking about antibiotics. So it produces a poison that is called as an antibiotic. Now, what happens? This antibiotic, when it reacts with bacteria, the bacteria dies. Now, several generation of bacteria die. Now the bacteria understands, okay, the fungi has developed a poison, and now we have to fight back. How do we fight back? We are trying to build a character. Okay, we will try to build a resistance. Resistance means something that cannot let you die because of antibiotics. So what bacteria do is, in the next generation, they develop resistance. Okay, so this resistance character or their ability to withstand this poison or antibiotic is called as resistance character. So this capacity of this bacteria to resist for antibiotics that are produced by other organisms is called as antibiotic resistance character. So that capability of the bacteria do comes from your plasmid DNA. Not only that, it also helps the bacteria to undergo a kind of sexual reproduction which is called as conjugation. It helps them to survive in extreme climates, pick up more characteristics. It helps them to use alternate source of food. Okay, so there are several functions to that of your plasmid DNA. Understood? Yes. Okay. So what you have to understand, the genetic material in the prokaryotes is present in the form of nucleoid. What is a nucleoid? It is a double-stranded circular DNA, which is naked DNA without the membranes, is called as nucleoid. Okay. And then the extra chromosomal DNA that is present in the bacteria is called as a plasmid DNA. Okay. Next, other structures that are very unique to that of your prokaryotes is, you see, if the bacterial cells or if the prokaryotes are motile, that means they can move, they have this long filament like structures which we call it as flagella. Now the flagella is very versatile, means there are very different types of flagellas are there. See, for example, now if I take a bacteria, okay, the bacteria can have more than one flagella. Okay, for example, let's say we have a motile bacteria, it has one flagella, and another motile bacteria has two flagella, and another motile bacteria has two flagella but on the opposite side, and another bacteria has a bunch of flagella, okay, on both the sides. Okay, so similar to that, we have many different types of bacteria based on the number and the position of the bacteria. Some bacteria have it all over the body, so it depends upon how they how they have evolved and how many flagella they have. But what are flagella? Flagella are the motile organs, okay, which are extensions, cytoplasmic extensions. Okay, so they're extended from the cytoplasm, and they these are very very long whip-like structures. What are whip? You might have seen this whip, right, Chati? Okay, so whip-like structures, long whip-like structures. So this long whip-like structures, what happens is they don't wave like this, but they rotate, okay, like a motor. So they rotate like this. That will cause this entire bacteria to spin rather than move like a snake. Okay, so that's how the bacteria works. Like bacterial flagella works. So bacterial flagella, as I said, it's an extension from the cell wall, not the plasma. Sorry, it's an extension from the cell wall, and based on arrangement, based on number, we can classify bacteria. But if you see the structure of bacteria, structure of flagella of bacteria, you can divide it into three parts. Okay, the structure of flagella can be divided into three parts. What are those parts? One is a filament, other one is hook, next one is a basal body. Remember this. There are three important parts with respect to flagella. One is a filament, next one is a hook, other one is a basal body. It's very easy to remember. Now, if you look at the filament is a very, very long whip-like structure or tail-like structure which moves in rotation and helps in the motility of the bacteria. Now, if you look at this plasma membrane, if you look at the whip or the filament, the entire filament of the bacteria is covered by plasma membrane. Understood? Within this plasma membrane, you have the structures of the filament that supports them. We'll study later. What is the structure of the filament? Understood? Now, just below the filament is the hook region. The hook region actually connects the outer membrane of the cell wall, okay, to the exterior. So, see, this is within the surface of the cell. It does not come out, okay. So, it just projects and gives an anchorage for the filament to attach to. Okay, so that is called as a hook. Now, coming to the basal body, the basal body is a very very important part of the flagella. Now the basal body is made up of rings, and these rings are called as outer protein rings, and you have inner protein rings. Between these outer protein rings and inner protein rings, you have a shaft like a rod. Okay, so that is called as a rod. Okay, now what happens, this particular portion, the rings are present within the plasma membrane. Remember that the outer ring and the inner ring are present at the plasma membrane. Okay, they are present at the plasma membrane, and these rings, at the middle of the rings is a rod. Okay, so now what happens is these are called as protein motors. So these protein motors actually spin, okay, and generate this movement across the sleeve so that the motion is achieved. Understood? So this is about the structure of the flagella. There is more details about structure of flagella. We can discuss it as we move on with respect to the cross section of the flagella. Understood? So it's very simple. We have the basal body, we have the hook, and then we have the filament. The basal body is made up of rings, okay, and these rings in the middle we have a rod or a shaft. These are protein motors that are connected across the plasma membrane and as they move up to the cell wall. Now in the cell wall region, what you have the hook region that gives an anchorage for the basal body, or sorry, for the filament. The filament rotates and causes a motion that helps in movement of the flagellum. So don't worry, we're going to discuss more about the flagellum as we progress. Understood. So now you can see the some of the images of your flagella. Now, apart from this large extension, you have other extensions on the cell wall of the bacteria, and we call them as fimbriae, and others we call it as pili. Now, pili and fimbriae are two cell wall extensions that are very unique. Now, when we talk about pili, pili is a structure. Now, this pili, what it does is, let's say you have one bacteria, and then you have another bacteria. Now, one of the bacteria can survive very harsh conditions, and other bacteria cannot survive the harsh conditions. Now, what happens is these two bacteria come together and then they attach. So they attach together to through this cytoplasmic extension called as a pili or pilus. So this pili or pilus, it creates a bridge between the two cytoplasms, which we call it commonly as sex pili. So during the sex pili, the characteristics of this bacteria get exchanged. That means both of them become capable of surviving in a harsh condition, similar to that. So that happens. So that is the structure. That is a function of pili. Okay, pili is nothing but an elongated tubular structure which is made up of pelicin. Okay, or pilin is a protein. Okay, or pelicin is a protein. Next one is a fimbriae. Fimbriae are bristle-like, your toothbrush-like structures which are spread across the cell wall of the bacteria. Now, what is the function of a fimbriae? Fimbriae, they help in anchorage. You know that you have the bacteria in flowing streams, or the parasitic bacteria will have to anchor to the host tissues, okay, etc. So they go and attach to the rocks and the host tissues, and then they help in attachment. So this is about flagella, pili, and fimbriae of the prokaryotic cell. Understood? Apart from these structures, which are very unique to the bacteria, and the next structure is ribosomes. Okay, give me a second. Yeah, it is a ribosomes, and we will discuss about ribosomes in the coming session. Did you guys understand? Yes or no? Students, are you able to follow? Please let me know.